I'll start with a welcome to the 2020 Dairy at Guelph Virtual Showcase here at the University of Guelph. My name is David Kelton. I'm a faculty member at the Ontario Veterinary College, and I have the privilege of serving as the Dairy Farmers of Ontario Research Chair in Dairy Cattle Health. I'll also be the moderator for the session this morning. This is day two of what is actually our fifth annual showcase event since we informally founded Dairy at Guelph in 2016. Unfortunately, we can't have an in-person face-to-face meeting this year as is our norm and our practice in the past, so we don't get to meet with all of you face-to-face and one-on-one. But I think one of the perhaps a bit of a silver lining in the COVID cloud is that this does, I think, provide an opportunity for those that are further away from Guelph to spend some time with us without having to travel here for the event itself, and so we perhaps can reach a bit of a broader audience in this way. So let's look at that as a positive. This is the second of the 2 days of our virtual event this year. We started yesterday with a couple of keynote speakers, and then today we have a panel of 5 of our outstanding researchers here at the University of Guelph. Each of our panelists has pre-recorded a presentation, which was posted on our website, and as registrees for this, or registrants for this event, you will have had access to those, and hopefully you've had a chance to view those. Our plan today is to move directly into an open question and answer session with our speakers. In order to do that, what I will ask you to do is to use the chat box, which you will find at the bottom of your screen, and to post your questions there. You have a couple of options. We'd like you to post to all participants. That way that our panelists can see the questions, and I, as the moderator, can see the questions as well. Our host for this WebEx broadcast is actually Dan Lacroix, and Dan is our technical guru and the person behind the scenes at Dairy at Guelph who does all these things, but he doesn't necessarily want to see your questions. He's running things in the background. So please post to all participants. And if your question is directed to a particular panelist, please indicate that in your question as well. So before we get started, a couple of things. Again, I want to thank Dan Lacroix, who is our administrative support for Dairy at Guelph and is working behind the scenes. Thank you very much, Dan, for that. And the second is, for those of you who were with us yesterday, have heard this already, and I promise I won't play the video because that was a technological disaster and shows off my technical inabilities. But we founded Dairy at Guelph in 2016 as a somewhat informal gathering of and group of 60 to 7 faculty from across the university engaged in, to some degree, in dairy research. We've done a lot of things in the last 5 years, and we've reached another milestone very recently. We're very pleased that the University of Guelph Senate at their October meeting officially recognized Dairy at Guelph as the Center for Dairy Research and Innovation here at the University of Guelph. There is a short video announcing the launch that is available on the website in the same section where you perhaps previewed the panelist presentations today. I won't try to play it. As I said, that was a bit of a disaster yesterday, but I encourage you to have a look at that. Nonetheless, as a group, we're very excited because I think this just offers us another opportunity to perhaps do an even better job of reaching out and connecting with our stakeholders and all of you out there in the dairy industry. So we're very excited about that, and we look forward to the steps now in transitioning to that formal center here at the University of Guelph. So at this point, we'll move to our panel session this morning. As I said previously, we're very pleased to have 5 of our leading researchers here at the University of Guelph with us this morning. I think most, if not all of them, are familiar to you, and they've all shared with you a presentation of some of the work that they've done in research recent times. I will give a brief introduction to all 5 panelists here at this point, and afterwards I'll give each of them just a minute or two, perhaps to make some opening remarks, which may be some high points or key points that were included in their presentations or other remarks that they may want to make. So our panelists, we'll start with Christine Bays, who's an associate professor in the Department of Animal Biosciences and also the Canada Research Chair in Livestock Genomics, and her presentation was entitled Genomic Selection for Health and Efficiency. Our second panelist is Stephen LeBlanc, who's a professor in the Department of Population Medicine, and his presentation was entitled Prudent Use of Antimicrobials. Trevor DeVries, professor in the Department of Animal Biosciences and Canada Research Chair in Dairy Cattle Behavior and Welfare, and his presentation was on precision feeding and management. Michael Steele is an associate professor in the Department of Animal Biosciences and NSERC Industrial Research Chair in calf management, and his presentation was entitled New Concepts in Calf Nutrition. And finally, Giselle Lapointe is a professor in the Department of Food Science and the Dairy Farmers of Ontario NSERC Industrial Research Chair in Dairy Microbiology, and her presentation was entitled Adding Value to Dairy Products. So at this point, probably in the same order as I introduce them, I'll ask each of our panelists if they have some opening remarks that they'd like to make, and we'll start with Christine.
Thanks, Dave. Hello, everyone. Greetings from Maryhill, Ontario. I hope you've enjoyed our presentations, and if you haven't had a chance to look at them yet, please do so. My colleagues and I have we hope to have given a nice cross-section of the work that we do, and we can use this platform today to answer any other questions you might have. Feel free to contact us afterwards as well. While recording the presentation or preparing the presentation, there were a few things that occurred to me. Number one, that I'm extremely grateful for the students that I have the privilege of working with. Number two, that I'm so grateful to do my research at the University of Guelph, where there is just an incredible group of top level researchers involved in dairy cattle research. I've benefited so much from the knowledge and expertise of Dairy at Guelph faculty and researchers, and I really look forward to continuing and expanding my research with them. And finally, I'm also very grateful for our Canadian industry and the funding agencies. Without their support, data, and interest, we wouldn't be able to do really anything at all. So also a big shout out to those stakeholders in Dairy at Guelph. And with that, I look forward to an engaging discussion with all of you about the exciting lineup of research that we're doing at Guelph and across Canada. Thanks, Dave.
Thank you very much, Christine. Stephen, some opening comments from you. And you may want to unmute yourself.
There we go. Thanks. So, yeah, the presentation that I shared was a brief synopsis of some of the work that collectively we have ongoing related to antimicrobial stewardship, and to pick up a little bit on some of what Christine said. I think it's a really nice example of some of the synergies that we find with the cross-campus group at Dairy at Guelph. Right now in Canada, we're not under the same degree of legislative or social pressure to cut antimicrobial use as what's happened to some of our colleagues, say in the Netherlands, for example, or in other countries. But it's certainly true that the amount of scrutiny and eventually the amount of pressure to cut use and to be seen to be doing our part as an industry towards the global problem of antimicrobial resistance is only going to grow. And so we're trying to get out in front of that on a number of fronts. And so there are people at the University of Guelph who are working very much in the microbiology of these things and molecular mechanisms and so on. Others who are providing advice to some of the UN level boards and bodies that are trying to formulate policies and recommendations. But some of the stuff we have going on right now that's I think really leverages our strengths in cross-cutting areas is actually doing some social science type of work where we're doing focus groups and surveys to really get a better handle on the decision-making processes that go into how farmers make treatment decisions, so that we can be out in front of policy making and have that be informed by what end users are actually thinking, what their preoccupations are in terms of animal health, animal welfare, and animal performance as we evolve through how we maintain health and use antimicrobials to that end. Looking forward to today's discussions.
Thank you very much, Stephen. Trevor, over to you.
Yeah, well, thank you very much for the opportunity to be part of this panel today and to share with you all some of the, I guess, a summary of some of the work that we've done in this area of precision feeding and management of cattle. And I guess in addition to what was included in my presentation, I guess one of the things I'd like to highlight is the fact that we've seen, and I did allude to that fact, is that we've seen a high adoption of technology in the industry. And as we move forward in the dairy industry, we're seeing more of that. And it's interesting to note, because we've done other work looking at not only obviously the functionality of some of these technologies, but even reasons for adoption. And dairy producers themselves will admit that a lot of technology adoption comes to them in effort to improve efficiencies, improve labor efficiency, improve the quality of their lives, which I think is really important. But at the same time, and that's some work that we've done as well, that has demonstrated that they're doing it in light of actually improving the quality of their cows' lives as well. And so that's where a lot of our research focus has been on, is looking at those opportunities, kind of win-win situations for the producers, but also for the dairy cows on their farms. And I'd like to highlight specifically some of the work that I highlighted was work funded through our Food from Thought program here at the University of Guelph, funded by the Canada First Research Excellence Fund. And not only the work we're doing related to, as I mentioned, feeding and health management in dairy cows, we also have other work in precision nutrition being done by, say, our colleague, Dr. John Kant here in the Department of Animal Biosciences, very similarly looking at opportunities at a cow level to improve efficiencies, target nutrient consumption in those animals, reduce environmental outputs, and overall improve farm sustainability through that research as well. And so, again, I look forward to some of the discussion that we might have on this here today.
Thanks very much, Trevor. Appreciate that. Over to Mike Steele.
Hi. Firstly, I'd like to thank everyone for this opportunity to be a panelist at this event. I'm really excited to share some of my research that's in the pre-recorded session, which I hold and is predominantly from an NSERC Industrial Research Chair that I moved from the University of Alberta to the University of Guelph 2 years ago. Largely started by the Western Milk Boards, but then also when I moved to Guelph through support by Bayer, now Elanco, and also Trow Nutrition, which allowed me to continue this industrial research chair, which is completely focused on calf nutrition. So the areas that we research in our lab is predominantly colostrum management. So we've been looking at extended feeding of colostrum and the impacts on gastrointestinal development and health. We've been also looking at all the bioactives within colostrum outside of just immunoglobulins, from carbohydrates, lipids, as well as new proteins that we're identifying. And we're trying to figure out how they work in the calf and how they affect gut health, but also metabolism. We also study milk feeding a lot, so different levels of milk feeding and how that imprints the calf, and also now the composition of milk replacers and determining why the macronutrient composition is different and how that can impact the calf's health and also performance. We also do a lot of weaning studies, and we've been looking at delayed weaning and step-down protocols, but in addition, the composition of solid feed during this time and how we should be tailoring the solid feed composition to the milk or milk replacer feeding scheme. So that's just a little bit of the research that we've been doing related to calf nutrition. We're also very interested in alternatives to antimicrobials and recently fetal programming. So very excited to be on this panel. I look forward to the questions. Thank you.
Thanks very much, Mike. And Giselle, over to you.
Okay, thanks. I add my thanks for the invitation to participate today. And I guess what I would like to add is that there's a lot more happening in dairy on the food side, especially with regards to dairy proteins and fats. Dr. Doug Goff has been looking into how the level and type of dairy proteins provide the feeling of fullness or satiety when included in a breakfast meal, and whether this can influence insulin levels and reduce calorie consumption at the second meal of the day. Dr. Shai Barbut is using dairy proteins as ingredients for meat formulations, and he's looking at how they can provide unique texturing and binding properties. On the other hand, Dr. Marangoni, who some of you might have heard speak about fatty acids, is studying how we can improve the nutritional and physical functionality of foods. And his expertise is in the physics of food materials. And how do you keep lipids solids for uses such as shortening while you reduce to trans or saturated fats? And finally, Dr. Art Hill is in full swing leading the development of online courses for dairy and cheese technology, for example, in the Master in Dairy Technology and the Cheesemaker Certificate Program. And we should be seeing those becoming available in the summer of 2021. So I'm happy to be here to answer any questions you may have following the presentation.
Thank you very much, Giselle. So we are ready now for our Q&A session, and I see the chat box at present is empty. Folks, we've got a nice audience out there. I'm sure you've got some burning questions for our panelists. That's why we're all here. Please don't be shy. Post
There's one in the Q&A from Raymond.
Okay, I'm not.
Transition milk.
Okay, I'm not seeing that.
Maybe underneath the chat.
Okay. Oh, all right. Yeah, sorry, it's sort of scrolled off. So, question here: transition milk. What feeding rate would you recommend from days 2 to your choice to improve growth and health? And so I suspect this is probably to Mike.
Sure. If you can pool your transition milk on farm and feed it to the calves after their second meal, so they should at least receive 2 meals of colostrum, then follow that with your transition milk. If you're pooling the transition milk from your cows, I think it's okay to feed it after those first 2 meals for that period. However, a lot of people are mixing colostrum and the either pasteurized whole milk or milk from the bulk tank or a milk replacer now. And what I would be doing is trying to mimic the total solids that would naturally happen in the first to the 5th milking in a cow with these colostrum replacers or colostrum to milk ratios. I think that's the best way, is just to follow what naturally happens in animal, in a lactating cow. Now, there's other ways of doing this too. That could be too complicated, but essentially there's a lot of people freezing colostrum, putting ice cubes into milk or milk replacer, these colostrum ice cubes, but there's many different ways of this. But I think the key take-home message is that you should have a gradual reduction from colostrum to a milk or milk replacer over the first 3 days of life.
To follow up on that, Mike, just like you said, you could take the colostrum itself and use that as kind of a dilution. Is that worse than the actual transition milk itself? Because colostrum is obviously different than the transition milk itself, right? So even though it is kind of a diluted form of colostrum, it's not going to be the same, right?
Yeah, so some of the bioactives will be the same. Obviously, the immunoglobulins will be a lot higher compared to transition milk, but some of this transition milk, and the more we characterize it, we're realizing that some of the bioactives that are high in colostrum are still high in that transition milk, like some of the hormones, some of the prebiotic oligosaccharides too. So we think it's an acceptable way if you can't pool on a farm your transition milk, but my first choice would be feeding the exact transition milk from that cow or a pooled transition milk that you're collecting on farm before going into these situations where you're mixing colostrum and milk or milk replacer, just because there's more opportunity for error when you do that.
Thanks, Mike. Maybe Trevor, a question for you. Certainly we've seen quite the evolution of technologies around milking, and certainly your presentation. You've been involved in a lot of that, not only looking at the technology, but how cows interact with that technology. What do you see on the horizon in terms of sort of what's next? We've seen some tweaking, some evolutions in the technology. What is your sense of where either the industry is going or where perhaps they should go in terms of this automated milking system technology?
Yeah, good question. And I was formulating an answer, and then at the end you said go in terms of the automated milking technology, right? So I think we've done a pretty good job, and the industry's come a long ways when it comes to the automated milking technology itself. And I know there's been attempts, and there is that technology out there to say automate parlors as well, right? And that's being done primarily in the spirit of labor reduction, not necessarily from a cow behavior, cow health and productivity standpoint. And I still view the future when it comes to automation in terms of milking as being single box kind of robots where cows are milking voluntarily on their own schedules, rather than say automating existing systems that we might have. And again, I'm not sure. Like I don't think I've let my brain kind of creativity flow enough to think of whether or not there's the next step there as far as how we might further automate that. And again, just over the past, say, almost 20 years, or almost, 30 years, that we've seen automated milking within the industry, we've seen the technology improve that much, and just tweaks to those systems. Again, there's probably going to be some revolution in that as well, but I think the integration of those systems and other technologies within the barn, whether that be feeding technologies and other behavioral monitoring technologies and those kind of things, I think that's where we still have a lot of opportunity. We have a lot of these technologies working almost in silos, and the integration of everything, and there are people working on this, the integration of those things at the herd or farm level is probably where we're going to be going next.
Thanks very much, Trevor. Appreciate that. And maybe I'll sort of follow up with that and maybe ask Christine to comment, because I know that Christine, with the some of the genomic work that she's been doing and trying to collect phenotype data, she's really been at the forefront of trying to deal with some of the challenges with integrating data from a number of different platforms, in terms of what some of the more intensive data collection that she's involved in at our Laura Research Station. Christine, do you want to comment a little bit on that, about sort of what your sense is of where we are in that process, and what some of the challenges and maybe some of the successes have been in that term?
For sure. It's a really exciting time. And I think all of us researchers for sure benefit from these technological advances that we've seen in the past decade. We're at the stage where we can now use automated technologies to actually help out with breeding programs, which is sort of unthinkable, or it used to be unthinkable. Elora in particular is a special research station because it's so fancy. There's so many things that we can collect there, and that we are collecting there. Our cows, we're trying to limit the amount of jewelry that our cows wear in terms of collection, but we still want to have the most comprehensive data collection on live animals. And in particular, it would be really great if we could collect data from conception all the way through to the end of a productive cow's dairy life, or life on the dairy. Some of the benefits, I think, of doing this is that we have the opportunity to really, first of all, integrate the knowledge of all of our dairy researchers at Guelph through this facility, because we're all measuring things on the same cows. And I think that puts us in a really good spot to be able to not only do our own research, but also break down the silos that have existed in the past, and and really share cows and share information and knowledge to try and advance dairy knowledge as a whole. It's gotten so specialized that that's sometimes very difficult. You really have to switch your brain to be able to speak with an ethologist or a nutritionist as a geneticist. But I think that that's where we have to go. So that's an advantage of these technological advances, because we can, if I'm looking at the genotypes of an animal, I can actually speak with either Trevor or Mike or Eduardo Ribeiro or Dave or Stephen LeBlanc and talk about the phenotypic side that they're looking at. So that's really a huge advantage, and it's really exciting. A disadvantage is, of course, that we're all competing for the same cows. So if I want to do a very detailed, I have a very detailed research protocol at Elora, and someone else comes in and says, well, I want to use the same cows, it's a limiting factor a little bit. That being said, we've managed to do a really good job of splitting the research resources amongst us, and it's really exciting actually, because it forces us to work a little bit more closely together and to really think about how these experimental designs can fit with one another. On an industrial standard, there is just a huge push to get these automated sensor technologies into data pipelines that will be useful for the industry as a whole. And I think that we're not there yet, but we do have to keep on it, and we have to keep at it with organizations such as Lactanet, even international organizations such as ICAR are really trying to standardize phenotype collection so that we can trust these individual systems. We can work with the companies that are producing them, such the Lely's and the Gias and these types of companies, in order to really standardize the data that's coming out of them and to make it useful for much larger things like genetic evaluation on an international scale. Does that answer your question, Dave?
Perfectly. Thank
Wonderful.
you, Christine. Very much appreciate that. So it looks like people are finding the chat box, and so we're starting to see some questions roll in here. And so again, folks, please don't hesitate. The panelists are here to answer your questions. There's one here from Diego Nobrega, and it's directed initially at Stephen LeBlanc, but he suggests that perhaps Mike and Christine might want to comment afterwards as well. And he says, during your presentation, you commented on reducing use of critically important antimicrobials in dairy herds. I'd like you to comment on the availability of alternatives for dairy producers, which includes non-antibiotic approaches and also non-critically important antibiotics, and also comment on any ongoing research in that direction.
Yeah, so the critically important, meaning of the highest importance for use in human health, relative to the dairy industry, largely we're talking about ceftiofur, which is sort of this paradoxical, very high importance to human health, and therefore something in principle we would like to limit the use of. And yet it occupies a very particular niche as the only antibiotic that has a labeled 0 milk withholding time. And so on the one hand, it's labeled for a number of uses, and its withdrawal time makes it an important tool in the toolkit with respect to residue avoidance as well. But really, that's the drug in particular that's attracting the most attention in the dairy industry. As an interesting aside, that drug itself isn't actually used in people, but to the extent that there may be resistance of bacteria against ceftiofur, that tends to bring along resistance to a number of other drugs, which are some of the big guns that are used directly in humans. In terms of alternatives, there's a small but growing body of work that would indicate that some other antibiotics, in particular to treat metritis, which is one of the common use indications in dairy cows, that there are alternatives which would be of somewhat lesser but not low importance in in human medicine. So really, the path that we're hopefully going to go down next year is kind of on two prongs. One, to circle back to what we've been talking about a minute ago, is to use some bits of sensor technology, activity, rumination, visits to a milking robot, some other streams of data coming in from precision tech that might help us to identify cows with botrytis or other health problems earlier. And that may allow us to use therapies for those animals that are non-antibiotic. So for example, with anti-inflammatory drugs, and we have some options there that are labeled for use. And again, there's some work that's been done in Europe to indicate that might be a viable path. There's also a little bit of emerging work from the States to say that there's probably a subset of cows, again, with botrytis, just to use that example, but you could make the same case for mastitis, that probably don't need antibiotic therapy at all. And if we could just be smarter and more strategic about identifying those cows, be it with technology or just better predictors that people can observe, all of those strategies will probably help us find a way forward to minimize, if not eliminate, the use of those critically important drugs in dairy cows.
Yeah.
Thank you, Stephen. I'll maybe now invite others who may want to comment on that. Mike, I think maybe you have a comment.
Yeah, that's a big piece of our work right now, is trying to find alternatives to antimicrobials, because antimicrobial use in the calf world is rampant. So not only does 50% of calves get diarrhea, most of those get treated with diarrhea, with antimicrobials. So it's a big issue. So what we've been doing is focusing on sound nutrition first. So there's still some basic concepts, I think, in just macronutrient composition that could be setting up these calves for failure and possible disease. So we're trying to correct that. But we've been looking at alternatives to antimicrobials, mainly on the probiotic side. And we've had some success in the calf world with some Saccharomyces cerevisiae boulardii, where we've shown reduced incidence of diarrhea. And we've been studying more how these probiotics and microbial solutions alter not only the microbial population, but the host, so the calf, and how the health is changed through these responses. So that's been our focus, and it's been awesome coming here to the University of Guelph and working with health experts in OVC. And a lot of this work that I've been doing in this area is with Dr. Dave Renaud, which has been really amazing to collaborate with another dairy at Guelph person who's enabled us to look at larger populations with more ease, and it's been a great collaboration.
Dave, I have a follow-up question for Mike, if I may.
Absolutely. Sure.
Yeah, so you talked about Saccharomyces as a treatment, and I know that in your presentation you mentioned the prebiotics. So do you think that those prebiotics need to be combined with other types of probiotics, like is done with humans, in order to have a more efficacious, to have more efficiency against diarrhea?
Yeah, I think a lot of the probiotics being used, especially in the calf world, but even in other stages of life, actually work better when they have a prebiotic associated with them. For the way that we study things, we like to reduce things and just determine if it's the prebiotic or the probiotic or the combination having a synergistic effect. So we like to tease them apart a little bit before we start combining them. But yeah, that's a very common way, and a common approach is to combine these pre and probiotics. So
yes, but do you think that the prebiotics can be used more efficiently alone, that the components are already in the microbiota, or we do need to add other components, other microbial components as well?
All I can comment is that I think the responses are a lot more consistent with prebiotics. I think probiotics are more tailored to the environment or of the gut microbiome at that stage, but there seems to be a lot more consistent responses in calves with prebiotics. Yeah, and a lot more variability with probiotics.
Great, thank you. And anybody else? Christine, did you want to make a comment?
Yeah, I think it's a great question, and it's a great it's a great challenge for all of us, and it's a really great example of where we have people approaching the same problem from very different backgrounds. From the genetic side, there has been since the 90s a history of trying to include these really difficult traits, such as health traits in general, into a genetic evaluation system. We started with somatic cell score, which is a really one of my least favorite traits because it's such a terrible trait. It's not exact, it's not good, but it seems to, pragmatically speaking, it seems to get something done, which is good. We moved on to, through a lot of work from the industry, they've now included metabolic disease. Hoof health traits have been included, with phenotypes being collected by hoof trimmers. We're planning, or LactaNet is planning on including fertility disorders by the end of this year. And moving forward, we're trying to look more at this calf health problem. We've got a large research project that I mentioned in my presentation as well, where we're looking specifically at calf health traits, because at the moment we've got, between stillbirth and calf deaths under 2 months of age, it's a large percentage of animals that are not doing well. And we don't have any way, shape, or form of collecting enough data in a standardized way across the country to address this. And I think that we've done well with our genetic evaluations in the past, but we should be constantly striving to do better. And this is an area where we can do better. The recent, well, it's not recent anymore, I guess, but the large spread of the haplotype cholesterol deficiency, or the cholesterol deficiency haplotype, by a very popular bull. It spread like wildfire in Canada, and we didn't even realize it. We had to have the Germans come and tell us that we had a problem with one of our animals, with a multitude of animals, or that the world had a problem. Really, I think that these things can be avoided if we work together, and we have the technology now to do this really well. So I think genetics also offers an opportunity to address antimicrobial use and to provide the world with healthy animals.
Thank you, Christine. And maybe while I've got you here, Ashley Napton asked a question here earlier on that maybe it's a good time to circle back to. And so again, you talked a lot about evaluating health and longevity traits, for instance. You also talked about inbreeding in your presentation, I think, and so can you maybe expand on that? And Chas, when you touched on the impacts of inbreeding and milk yield potential, did you evaluate other health or longevity traits in that context? And what did you define actually as highly inbred when you think of inbreeding?
So Ashley is referring to a number of studies done by Biode Macagnola, one of my PhD students, who really is a fascinating person in himself. But he does this work, and he digs in deeply into these questions. So in Canada right now, we tend to use, we're still using pedigree inbreeding, and this is kind of missing out on a lot of variation that is, or a lot of inbreeding, I guess, or homozygosity that is real. If we use pedigree information, we're just kind of guessing how many genes come from each of the parents, and we're saying that we think that 50% comes from the sire and 50% comes from the dam, and we lose sort of what we call Mendelian sampling variance. And using genomic information, which has came around in the last 10 years, we can really exactly define areas that we can afford to be homozygous and other areas that we can't afford to be homozygous. Now, in general, a selection program will increase homozygosity. We're breeding related individuals together, and that has been what has propelled us into this huge productive era, really, of livestock, not only in dairy but also across other species as well. So inbreeding is not all bad, we have to say. We are currently in the process of looking at other health and longevity traits. They're a little bit more difficult because they're not well defined. It's very easy to look at the quantitative measures like milk yield and protein yield because we have real measurements. But when we get to fertility, traits, which we've looked at in BioDay's publication, it gets much more difficult. Health as well. We need to have really quantitative measures, but we're starting to collect those. And next year, we plan to continue this research and include more health traits. I think it would be particularly interesting to look at the calf health traits that we're starting to collect. And once we have those, we'll be able to do to answer that a little bit better and use genomic information to answer that. So we'll have really ultimately the true quantitative measure of inbreeding depression in some cases. What's defined as highly inbred? That's a huge question, and I'm going to try and put it in a nutshell. The measures that we've been using to define and explain inbreeding are old. We have livestock populations today which are being influenced by reproductive technologies, by artificial insemination, which has really revolutionized how we breed livestock. Now, the question, we use a measure called effective population size. That's a theoretical number of how many males and females do you need to maintain the diversity in a population. I don't think personally that measure is useful in today's world. We have gene banks. We have, our million cows in Canada are being bred to about a couple thousand bulls. This is totally different than the natural assumptions that would have been made to define something like effective population size. So it's hard to answer that question. We can have a long discussion about it. I would say that if you have an inbreeding increase, or the increase in the rate of inbreeding in a population that's higher than 5% per year, then you should be worried, even with a managed population. But an animal itself, its own inbreeding isn't so important. It's more the relatedness between individuals in a population that is going to affect the future inbreeding and the future diversity of that population. Sorry for rambling. I'm done now.
Thanks very much, Christine. Appreciate that. I've got a question here for Giselle. Giselle's been let off the hook here so far. We'll get her involved here, and this comes from Ashley as well. Giselle, are you looking at particular mycotoxins related to silage quality? Your presentation mentions ones that are a result of poor fermentation, but wondering if you're looking at field toxins as well.
Well, I gave the mycotoxin production as a very good example of silage quality. We're not looking yet directly at mycotoxins in either the crops or the silage, but we are interested in antifungal lactic acid bacteria. So I know when the mycotoxins are produced in the field, you have the mycotoxins in the silage, not necessarily the action of the molds in the silage. So that would require especially superbacteria that would be able to degrade the mycotoxins efficiently in the physiological environment of the silage as well. So yeah, it will be important in the future. Right now, we're trying to get a handle on the variability but we're not looking directly at mycotoxin level. We're looking mainly at the microbial context. So we're looking at the types of microbes that are present, and not yet what they're actually whether they're actually producing mycotoxins.
Thank you. Here's an interesting one. So Warren Skippen sent me a private question here, and it circles back to calf raising, and we got into this in yesterday's session a little bit because we talked about cow-calf separation and sort of leaving calves with cows longer. We've certainly seen some changes in our industry from sort of moving from individual calf housing to group calf housing, and sort of prolonged, in at least some parts of the world, particularly Europe, are actively looking at keeping the calf with the cow in some format for a prolonged period of time, likely until weaning. And a couple of questions, and maybe to Trevor, from the perspective of, are there any potential concerns or impacts that you can think of in terms of then these animals ultimately adapting to other technologies such as robotic milking? And the other part of that is to maybe get some comments from Mike in terms of, again, some of the impacts on sort of that type of a transition in terms of early calf nutrition and so on. So maybe Trevor, we'll start with you.
Yeah, no, good question. And I think I saw the question too, like it's around not only the potential of, say, extended maternal contact, but also grouping calves, right, and housing calves. I actually was on a meeting yesterday where I got a very similar question in terms of adaptability of, say, heifers to robots, and yeah, does early life management influence that? And I would like to say yes, obviously, or not obviously, but we don't have any empirical data to say that's the case. Lots of anecdotes about these things, and lots of suggestion in the field that exposure to, say, technologies early in life, as well as in the period leading up to, say, introduction to a robotic milker, for example, would have a positive impact on the transition of those animals. Some of that work that's being referred to and mentioned there, both say extended contact time with the dam and or group housing, right? Pair housing even from an early age would suggest improved cognitive development in those animals. A lot of work being led out of UBC on that topic area, looking at that, and part of that is behavioral flexibility, and so not only willingness to adapt to new feeds, new environments, new social environments, but better ability to react to changes as well. And so we could make the leap there probably that that's going to have an impact on those animals, especially if that's reinforced early on life into those animals as they transition to, say, on a robotic milker. And I guess that's where, and in my presentation, I highlighted some of the work that we've been doing. One of my grad students, Anna Schwanke, has been looking at personality of cows and how that relates to robotic milking. And what's interesting there is that it's not only, say, fearfulness in the case of the couple examples I provided there, fearfulness to novel objects, which would be speak a bit of, again, to the adaptability of those animals to, say, technology or to novel situations, but also the human interaction part there as well. And the fearfulness to humans having just as big of an impact, maybe even greater. And that's something we've seen as well. Like exposure to technology and all these kind of things is great, but I've seen situations where those animals almost become too reliant on technology and not enough reliant on humans in terms of having that interaction with them. And when we lose that interaction or piece there as well, then we've seen challenges there as well. And I've seen robot barns actually where animals come in that have had very little human interaction, kind of like an extensive beef animal, where that actually poses more challenges. So, but coming back to the original question, both those things are going to be highly influenced by the management of those calves early on in life. And I think that's where a lot of this research has shown the positives of that as compared to this kind of traditional separating calves immediately, weaning them or rearing them on their own without or very little social contact of other animals. And that has been shown to have kind of negative effects on that behavioral development.
Thanks, Trevor. Mike, over to you.
Yeah, we've done a little bit of work in this area, not related to behavior. Mainly, our concern was if you're separating a calf from a dam, you're not going to get as much exposure to a more mature microbiome from this dam, and the exchange of the microbes is not going to be occurring. And our question was, does that affect the gut microbiome of the calf? And some preliminary work has showed that calves separated or calves with the dam, there's actually no difference between these groups, but this is very preliminary. So this is in contrast to infant research where you'd show big differences through separation. but still just the beginning. We've also seeded young calves with mature rumen fluid. So we're taking rumen fluid from a mature cow and seeding it in these calves that are raised in isolation. And again, we were able to change the microbial community a little bit, but no differences in health so far. So that's our interests, really just preliminary data at this point. We're just submitting it right now, but yeah, not a lot of differences from that context. But from a behavior standpoint and a feed intake standpoint, when you're pairing young animals in group housing, I think it's very well established that they're going to be consuming more starter, learning how to consume more solid feeds earlier, and have better weaning transition because of that. So I think there's huge benefits to group housing, especially calves, and also that coincides with automation and feeding using automated feeders, which I think are really untapped in our industry. And I think there's great potential to exploit them even further.
Great. Any of our other panelists want to weigh in on this at all? All righty. So I just want to get caught up on some questions that have come in, and some of them are, so Ray Rainen posted a couple of questions to Trevor. Feed tables in AMS systems and subacute ruminal acidosis. What is an ideal fresh cow feed table to balance production and health?
Well, I guess I could email Ray after with really specific numbers if he wants. But I guess, and I tried to make the point, I guess, is that we do have some challenges there. One of those challenges is actually the fact that, again, the amount of nutrition that, say, a cow will get in a robotic milking unit is going to be related to their visit frequency to that. And so something I didn't touch on, and I think that probably the first limiting factor is actually getting those cows to milk frequently in early lactation. And we have lots of really good old research to suggest that there's benefit of, say, increasing milking frequency in early lactation, and that's going to help proliferation of the milk secretory cells in the udder, and actually improve overall peak milk and overall lactational performance of those animals. And so that's where we have a huge opportunity. And so part of that challenge of getting enough nutrients into those animals is also getting those cows to the robot often enough in early lactation. And I think that's where we're often failing some of these cows. Not that we're not, say, our feed tables aren't set up for enough calories into those cows in early lactation, but they're actually not getting to the robot enough. Let's say we do get that fixed. The question is, how do we then minimize the risk of acidosis? And again, if the cow is visiting 4 times a day and we are providing not a huge meal of concentrate at each of those visits, the risk shouldn't actually be that high. Now, that is the most high kind of time period. And so that's where I think there is still opportunity for potentially feeding those cows a little bit different. I alluded to some of the work that we've been doing targeting, say, sugar supplementation. We've got an upcoming project looking at alternative kind of gluconeogenic precursors that we can give to cows in early lactation as an alternative to, say, getting more starch into those animals at that time period. But then part of that is, and the caution there is, we have a lot of variation in terms of production. We don't want to hold cows back either. And that's something that we've seen in the industry as well with our robots, is that if we don't get those cows onto a feed table quick enough that's based on production, we're going to end up either holding cows back or, say, potentially putting cows into negative energy balance as well. And so there is a balance there, and it has to relate back to then also, like I mentioned before, their milking frequency and all those things, that kind of that fresh cow protocol in those animals at the same time.
Great. Thank you, Trevor. Appreciate that. Question for Giselle. And this is a nice sort of segue here from, or tie into yesterday. Yesterday with our panelists, we were sort of looking at a broader perspective, both international and sort of more local, in some of the impacts of COVID-19 have had on the industry and on consumer behavior and so on. And there was certainly some discussion. Mike von Massow talked about, in changes in consumer behavior, more baking of bread at home, and that was certainly one example that he used. In another conversation, somebody said, yeah, we've been trying to churn our own butter at home or do some other things. Giselle, any comments, anything that you've heard in terms of other interesting things people are doing with dairy products, home yogurt production, any types of those types of things? Any experiences or thoughts about that?
Well, yes, actually, the attempts are very diversified. So I would say that it's even getting to making your own tempeh, making your own kefir. There's a lot of interest in fermented foods. And that was one of my questions for Mike, was about, he talked about microbreweries and that sort of thing and milling. But I also see a lot more interest in fermentation. So there's a lot more interest in diversifying, whether you're fermenting vegetables or fermenting dairy or whatever. Yes, there's home experimentation in those areas.
What are your thoughts on that as far as food safety issues or anything around that, or resources for people that might be interested in?
Well, there's some good resources out there, mainly the ones that are actually providing the starter cultures. So they have some good guidelines to keep safe on those type of fermentation. And I guess we're seeing a move back to many years ago. Home fermentation was a common thing, and there were cases of botulism, but I guess those we'll have to wait and see whether those will increase or not. These new attempts, but I believe the resources out there are very good quality, and hopefully people will be able to keep safe.
Thank you. Oh, we've had a question come in here from Bethany Muir for Christine. What is your opinion on engaging with nucleus herds for full spectrum quality records versus industry-wide voluntary participation in data recording, which needs significant edits and censoring in order to collect sufficient data for analysis of health, reproduction, and efficiency?
Yeah, great question, Bethany, of course. Yeah, we've seen, okay, so there are advantages and disadvantages to both of the approaches. Up until now, Canada has been really fortunate because our producers have willingly paid to have their information collected through registration, through DHI, through all sorts of different venues. In fact, they've actually also started collecting their own data on things like metabolic disease. We rely on these groups to collect data. And it's worked out really well because everyone has played nicely and contributed, and everything is all rainbows and butterflies. We're getting to a stage where that's not the case anymore, where indeed some farmers don't see the value of these data collection systems. That genetic gain that is available through those systems has been shared back with the industry as a whole, but it's kind of a I don't want to use this word, but I'm going to use it, a socialist system where everybody contributes equally and tries to get the benefits and spread them equally. We've seen something very different in the poultry industry and in the swine industry, where indeed they've moved towards nucleus flocks. The breeding organizations collect very detailed and high quality information from a few select farms, and that is what the breeding values are based on. The advantages to that are the quality of the data is, of course, impeccable. It's beautiful. You have everything under control. It's great. The limitation is that it's so great that it doesn't represent what happens on a commercial farm anymore. That's one disadvantage. The second disadvantage is that that approach limits your diversity. Again, we spoke earlier about inbreeding and that the fact that the effective population size of the Holstein breed is about 60 animals. If you look at it genomically, we don't have much diversity to play with, but we're still kind of trying to figure out how much we actually need. The poultry industry, if we compare dairy to poultry, they've sort of gone along the path of line breeding. It's foreseeable, and I think that is indeed the way that the industry is moving. Whether or not that's a good or bad thing, I think we still have to do the economic analysis. We have to find out how big our reference population should be, and we have to better understand the implications of collecting phenotypes on really well-managed herds. The poultry industry has gone so far as to actually employ commercial herds that don't have really nice environmental conditions in order to have some kind of an infection pressure in their animals. I don't know of anyone who wants to do that with dairy cattle, but it might be thinkable in the future.
Thank you. Appreciate that. I'll remind our audience that the chat box is where you should post your questions, please, and we'll make sure that the panelists will get around to them. Oh, here we go. Here's one for Stephen. He's been quiet here for a bit, so we'll give him the stage. And the question is from Warren Skippen. Can you give us a taste of what your qualitative producer interviews on antimicrobial use have revealed? What is their level of understanding of the need to use antimicrobials prudently?
Yeah, so I'll start with the caveat that this is very preliminary. We were able to get 7 focus groups completed just before we all went into lockdown in March, and that work is just about ready to go out the door for publication. I'll jump ahead and say that that has kind of teed up the next phase of that work, which is a very large industry-wide online survey questionnaire that we hope will be going live here very, very shortly to collect a broad section of further data on producers' attitudes, opinions, and practices. But to answer your question, we found, I guess, a couple of interesting things. Perhaps unsurprisingly, producers bring in a number of streams or threads of influences and information into how they make their decisions around antimicrobial use. Our work from these focus groups would say that there's not a high level of sensitivity or awareness or perception that antimicrobial resistance is a present problem in dairy animals, and that's probably an accurate perception. Further, there wasn't, from what we gleaned, a strong sense that antimicrobial use in the dairy industry or on anyone's own farm, sort of from their own lived experience perspective, that was an important thing, or that the connections from what might be happening on a given farm were obvious as to how that might eventually connect to public health and resistance in people. And again, that may well be accurate, but we don't really know. That's a tough thing to quantify. That being said, when we probed a little bit, there was, notwithstanding that, a pretty high level of willingness to engage with the topic, to think about ways that producers might reduce their own antimicrobial use on farm. And so it's not like there was an unwillingness to think about that or engage with that. And where the producers' thoughts went typically was very much towards prevention. They identified that having excellent facilities, good ventilation was probably what they perceived to be one really important piece of the puzzle. So I think on the upside, that notion of moving towards prevention as opposed to even just using, if you will, a lesser antibiotic as opposed to a more critically important one. So I think there's receptivity there, and again, that's that was deliberately a first step, and we're now going to go into a second step, which is to get a much larger sample and drill more deeply into those attitudes. And then a third step, and certainly not a last one, is that the plan is next year we'll actually test drive, pilot test some interventions that will be informed by what we've learned in those first two steps.
Great. Thank you, Stephen. Looking back through, here's a question for Trevor. And I think this comes from your pre-recorded presentation. You showed increased variation in concentrate intake was observed when concentrates were increased, but overall dry matter intake variability was similar. Was there a difference in daily milk production variability between the two groups?
Good question. And I saw the question, and I was pulled up my SAS just to verify something there, because it's not something we reported actually in the paper. But the short answer is no, there wasn't actually more variation. Now, again, this is a kind of a within cow trial over a fairly short period of time, and this idea of potentially more variability in robot concentrate feed delivery with higher feed levels is not novel to that study, and that's been demonstrated across a series of studies we've done, as well as that have been done at the University of Saskatchewan in collaboration there. Now, that being said, just because we didn't see that variability in milk production doesn't mean that it couldn't manifest itself. In other work at looking at variation, day-to-day variation in TMR, so complete ration dietary composition, we've actually demonstrated negative kind of associations between that variation, day-to-day variation, and overall herd intake and production. And so, again, I don't think it would surprise me if you would see that over a longer period of time and a greater kind of sample size. And I think it's just one of those cautions that we have to be careful of. And especially when using technology, we make a lot of assumptions. We say, well, the animal's programmed to do this. Well, we actually verify that they're doing what we program the system to either deliver or milk the cow so many times or whatever it might be. And so that comes back to our ability to manage some of that information as well. And I think that's part of the take home there as well, is we can't just trust that obviously what we're inputting always necessarily manifests itself. And we need to make sure that we identify those potential bottlenecks and then deal with those things as well.
Thank you, Trevor. Here's a great question that comes to us from Harm Kelly, and I think a number of our panelists can respond to this. And the question is, what 5 or so on-farm measures would you love to see farmers record related to dairy animals prior to them entering the milking herd? And so, he lists some examples: birth weights, weaning weights, total fluid milk consumed prior to weaning, etc. But if you had to, because I think one of the challenges that we have is that we don't have a lot of consistency in recording some of this data, particularly on those replacement animals as they're coming through our on-farm systems. So if you had a wish list, what would be on your wish list? And I don't know who wants to start.
I'd love to start on this one. I think this is an awesome question, and it's something I've been pleading to farmers to do, is to measure more, especially with young livestock, because there's so many associations between growth itself, even in the pre-weaning period, and lifetime production. So I think at the bare minimum, and I'm just going to give you 2, is birth weight and weaning weight. I think that information should be done on every single farm. It's not done on most farms, but I think just understanding that metric is really, really critical. If I had to add more, I would add 1 at 1 month, then 2 months at weaning. And depending on the farm, I would try to get 1 right before breeding, just so that you can look at that window between weaning and breeding through the pre-puberty stage as well as the puberty stage. And then from gestation, I'd like to get 1 right before they enter the milking line too. And that's just a bare minimum, but it's really sad from a growth standpoint that most farmers do not know their average daily gains pre-weaning, post-weaning, and I think there's a lot of dollars in that, and that would be my wish list, just for farmers to know, spend 5 minutes per calf to know their average daily gain.
Thanks, Mike. I'm not detecting any passion there, by the way.
Yeah.
Can I add to that, Dave?
You sure can, Trevor.
Yeah, no, I would agree with Mike, and I would say, and I would kind of highlight then some measurements around breeding age too. We've done a lot of evaluations over the last number of years as part of the Dairy Challenge program, and there's a lot of focus often with our students on first lactation performance and age at first calving. And when we go back and look at breeding ages, one of the things that we see is often, say, variation in production and early lactation in first lactation animals is related to that age at first calving, but we see a lot of variation around that. And so you'll see animals that are bred at 11, 12 months having like 4,000 or 5,000 kilo ranges in production differences within the same age bracket. And I would probably ascribe most of that to differences in growth in those animals at the point of breeding. And so often producers will say, well, I breed my animal at this age, or I breed them at this height, or right, they have to meet this marker on the wall in my barn or be up to this on my chest or whatever it might be. But we could be a lot more accurate if we had actual measures of animal size, weight, even height in and around that time period. And we could probably do a lot better from a decision-making standpoint on breeding on those animals. And then also not only immediately for those animals, but then informing management decisions. And kind of goes back to some of the measures Mike wants as well. We're not only going to use those for individual management decisions, but also a broader how we change maybe strategies. And then the other is just good disease recording in those animals as well. And many farms, most farms have excess heifer inventories right now. And we talk about using genomics for, making some of those decisions as far as keeping animals, et cetera. Probably one of the most simple ones is just looking at disease records. Did that animal have more than one case of pneumonia as a calf? Well, if she does, the data would suggest she's not going to be as productive, regardless of what we do to her. So that's an easy decision point. But if we're not recording those treatments, we're not recording those incidences, then that's a problem for us, and we can't use that information.
And if I could throw, I'll second and third that the disease records would be really useful and important. I mean, we alluded before that might be an important marker of the calf's future in things we haven't even really measured yet, but it's some of the past research done here and elsewhere would say that it's a bit of an insidious thing. I mean, fortunately, we've managed to wrestle down death losses from scours and respiratory disease to a reasonably low level on most farms, and yet calves that, for example, live through respiratory disease are about 1 in 5 of them will never make it to be a milking animal. And so if you look at the average milk production of your first lactation animals, that there's the variation that Trevor talked about, but those animals don't have a record, and so it's a bit of a biased sample, and it leads to us maybe underappreciating the issue and the losses. The other thing too, just to put in another plug, is antimicrobial use in calves is generally in the top 3, and we tend not to think about it so much because smaller animals, smaller dose, they're generally far from market, but actually it's one of the relatively important areas of use. And so again, for all those reasons, having good records on who got scours and who got respiratory disease both before and after weaning would really help us make some progress. And then last but not least, I mean, I think that's an opportunity for veterinarians, for nutritionists, for other advisors to lend a hand with just getting that job done, because doing the Whey taping and body scoring is not hard, but it's one more thing. And so if we can find a way to get that done, whether it's at Herd Health or when the nutritionist is making regular visits, maybe someday we'll roll that into milk recording related services, but there's opportunity there.
Mm-hmm.
Yeah, I've seen a lot of great progress in the field in this area, and now you see some vet clinics doing ultrasounds of lungs at certain points in every calf on farms. So it's really impressive how quickly that's developing.
If I add as well, Dave, sorry.
Absolutely. No,
this is when I need to...
I'd be disappointed if you didn't, Christine.
I need to agree wholeheartedly with everything that's been said. My list would contain calf health data for sure. Birth weight would be great. Weaning weight would be great. And with regards to all of these things, I think everybody mentioned that it's really important. We'd all be happy if producers would record this. But there's one step I think that we really need to underline. Recording it is not enough. We need a pipeline to get this information into an evaluation system that is standardized, that is useful, because otherwise it's just recording it and well, I don't want to say just, but people would use this for herd management. This data is so much more valuable and useful if we can get it together into a centralized system. I mean, a lot of things are being recorded already, but not being passed on to the evaluation centers. And this is really, again, I can only underline how important it is to pull the full value from all of this, because it's really a very competitive world out there. Canada has been traditionally well known for breeding really, really high quality animals. If we want to maintain that position on a global level, we need to step up and collect this information, but also make sure that we're using it to its full advantage. So I think that's one really important thing, that the pipelines are really important. And I just want to underline again something that Trevor said about, a lot of producers saying my cow has to be this high. I've heard the comment, cows need to be tall enough to breed. I hate to break it to you, but short people and cows get pregnant too. And they will never reach that height. So we need to sort of rethink what precautiousness is, or what puberty is, how we define it. And a lot of these things are known. It's just a matter of putting together the puzzle pieces and coming up with, a meaningful, sound, and strong procedure or operating procedure and data collection procedure that makes sense. We don't want farmers to be spending 12 hours of their day collecting data. They won't do it anyway. And that's ridiculous. But we do need to come up with something in particular with regards to this period that you've mentioned, Harm, this pre-calving period, because we're not doing a good job. We need to identify that, and we need to do better.
Thank you, Christine. Giselle, do you have anything to?
No, nothing to add on that side.
So I think our gold star so far for this morning goes to Harm Kelly for that question, because it certainly evoked quite a passionate response, I think, on from our panel, which is fantastic. So we are closing in on 11:30, and I think we'll sort of look to wind it down. I'm not seeing any more questions coming in. I think I'm going to circle back up to one that I think I missed. But before I do that, again, I will ask, once we've dealt with this question, I'm going to ask each of our panelists if they have a closing comment that they want to make before we sign off this morning. So panelists, you can think about that. And, oh, here it is. It was another question for Christine. And the question was, CEMEX is releasing a feed saved trait next week in the US. How does that differ from the work that is being done with the Genome Project in regard to feed efficiency?
Yeah, that's a great question, Ashley. And I've tried to answer it quickly in the chat, but I think it is really important to talk about this so that everyone gets the gist of it. there has been a huge push from the industry to put forward feed efficiency traits in the national evaluation system. There have been, I don't know how many meetings I've been in the past 2 weeks with the Genetic Evaluation Board, with the Breed Advisory Committee of Holstein Canada, talking about feed efficiency and how Canada is going to address this. The Americans, well, I guess I should talk about the project first. We've got a really big project that's coming up, and we're actually working together with the Americans, as well as a number of other international groups. Denmark, Switzerland, Spain, Germany, Australia are all involved, and we've come up with a way to collect feed efficiency phenotypes, and that is really feed intake on individual cows, cow weights, weekly or even daily, in some cases, DHI records, as well as some countries are measuring methane emissions. That's a whole bunch of background information. But the difference between what we're doing and the United States is doing is that we're using the same data, but Canadians are focusing on first lactation data. And what we've seen is that, what we believe is that it's extremely important to split the lactation up, because just one number with regards to feed efficiency over the course of a lactation cannot clearly define and show what's going on. So in Canada, we've taken sort of a pragmatic approach. We're using from 5 to 60 days as period 1, and from 60 days to 305 days of the lactation as period 2. That's the first difference between what we're doing and the Americans. We're only using the first lactation. The Americans are using multiple lactations. We're using a multi-trait model. So we've got correlations between that first period and that second period that are being included. It's a little bit more statistically demanding, computationally demanding, but we feel that it better represents the biological background of what's going on with feed efficiency. Another difference is that the United States is using a two-step genomic evaluation, We're using a single-step genomic evaluation. I know that those things don't maybe say much to you, but what it ultimately means is that the correlations between the Canadian feed efficiency trait and the trait that the Council on Dairy Cattle Breeding in the United States, which is the body ultimately responsible for genetic evaluations in the States, the number or the ranking even for bulls will not be the same. So we can don't be surprised if you see that it's not the same. They're not identical traits. What the Americans are coming out with in December will not be the same as what Lactanet will publish in April, but it shows that both countries are really pushing hard to include feed efficiency in a genetic evaluation. It shouldn't be a worry that they're not the same, because these models are different. And by combining the two, maybe some producers can come up with the best decisions for their herds. But over time, we, I think, are looking towards ultimately finding out what the best way to include feed efficiency into our genetic evaluations is. But ultimately, don't expect them to be the same, because they're not going to be.
Thank you very much, Christine. So we're reaching the end of, I think, our panel session this morning. As I said, I will give each panelist an opportunity to make some closing remarks. And maybe while you're doing that, or as part of that, Bethany Muir has posted one last question to all panelists. And she asks, are there any suggestions how we can, as industry organizations, more broadly promote Dairy at Guelph research and events? So, so if I'd invite all of you to perhaps comment on that. And I'll let Christine catch her breath. So maybe we'll start with Stephen for your closing remarks.
Thanks. And that was not a setup, but actually that's Bethany's question is a perfect segue to what I wanted to make my sort of parting words. You know, a couple of people at the beginning alluded to the fact that we all seem to be pretty happy at work and happy to be able to be part of this this collective effort that is Dairy at Guelph, and you know, at the risk of sounding a bit, I don't know, self-congratulatory or whatever, I do think legitimately our privilege and our good fortune is to be in a group of like-minded people who like to collaborate and like to be productive at the university. But not just that. You know, we have a great situation here in Ontario and across the whole country where I think most of us have an inclination to want to stay very connected to the front line of dairy farming. I mean, you know, hopefully we're all doing really good, rigorous science and publishing in journals and doing all those good academic things. But at the end of the day, the point of it is to be in service of farmers, of cows, and of the industry more broadly. And to do that, we rely on participation in our studies. Shameless plug for the farmers: when the survey comes to your inbox next month, please reply. And to Bethany's point too, it's a two-way street. So we will be very happy for spreading the word. We have started to do a number of, for example, one-page plain language summaries of the results of our research. There's a number of those up on our website, and there's more to come. As we get formalized as an official research center, increasing and improving our outreach through social media is one of the projects that we're going to work on. So being a bit of a multiplier for sending that out is really important to us. But equally important is hearing back from the front line. What questions do you have right now that are, hopefully we could answer with information from research, but also looking over the horizon. We certainly do that ourselves, but it will be a lot better when we're hearing back from end users as well, be that farmers or advisors, as to what questions and what preoccupations are on your mind for now and into the future, because that ultimately is what will guide our research.
Thank you, Stephen. Trevor, comments?
Yeah, I guess I'll say ditto to Stephen. I think he raises a very good point, and I really like this question from Bethany, because as researchers, we're called on to promote our own brand and our research, and we like to think we do that, and we have lots of opportunities for that, and obviously we're going to continue to try to do that as much as possible. One of the challenges I'm finding in the current kind of global situation we're in is we don't have in the last half year or so as much kind of that frontline contact as we traditionally would have. I love going to meetings, not only to talk about my research and some of the results, but also to have that one-on-one contact or group contact with industry to get feedback and think about where the industry is moving and going forward. And again, we're getting some of that, but it's just not as personal as it has been in the past. And so I think from an industry perspective, that's where I would really like to encourage, if you have ideas, if you have thoughts or feedback, to reach out to us as well, and especially as we kind of continue in this world of living and interacting distantly from one another. I think that's going to be important as we shape kind of the next couple of years as far as our research and priorities go as well.
Thanks, Trevor. Mike, over to you.
I think we're moving in a very positive direction, and I think the more partnerships with industry that we as academics can establish, not just with research, but also promotion of the findings and tech transfer, I think this is already happening. And I'll just use the event that I hosted here on campus last year, CAF conference. That couldn't have happened without industry support and guidance, actually. So I think more events like that. And it's okay if we're online for a period. Hopefully this won't last that much longer, but if we're forced to, I think some of these webinars and having active involvement in some of these webinars as we've had today is a step in the right direction before we can start meeting more face-to-face and participating in some of these joint conferences more so in the future.
Thanks, Mike. Giselle.
Yeah, I would add to Stephen's comments the consumer and consumer health, of course, as the natural extension with the diversity of expertise at Dairy at Guelph. And yes, I underline as well that we have to adapt to new online communication methods. and make sure that we're communicating and doing the tech transfer.
Thanks, Giselle. And now, Christine, hopefully you've caught your breath and we'll circle back to you.
I just talk too much. I think, sorry, I think that Bethany's question is great. I think that we really do have to strive to spread the word and increase discussion. And I mean, there are, I think at the hiatus, there was 45 participants in this meeting. There's no reason, we have just under 10,000 dairy farms in Canada. We could have had thousands of producers actually joining us here today. And it's free, it's good, and it's really interesting to just think about how other people view our research, if there are any comments. I think that moving forward, we should strive to have more producers interested in these types of virtual seminars. We've got 8 universities in Canada. I know we kind of came out there and said we're Canada's dairy university. We thought long and hard about that. And I think we can honestly say it's true, but that doesn't mean that we're on our own. We are constantly striving to include the best minds of everyone in Canada and across the world to do what we do best, and that is dairy research. And we're, as Stephen said, we're pretty good at it. We love it, and we would just love to continue to do better. And to do that, we need to spread the news. We need to increase dialogue and communication, and that will ensure that we can have a strong industry moving into the next decades.
Thanks, Christine. Thank you to all of you. Maybe I'll take a moment here and exercise my prerogative as moderator and make a few comments. And you know, we at the outset we celebrated this is the 5th year of this event, and really 5 years since since Dairy at Guelph sort of became this informal entity. Certainly one example goes back even beyond that, and it started over coffee with Milena Kordich, who was a faculty member in food science at the time, and she and I were having a chat about, you know, how do we get more engagement internally, externally, and do more of these good things. And I think we've come a long way in doing that. I think we still have a lot of opportunities and more growth. And part of that, as we went through the branding exercise for Dairy at Guelph, one of the things that we wanted to ensure, and the reason we called it Dairy at Guelph rather than Dairy at University of Guelph, was explicitly because I think we're more than just those of us at the University of Guelph. So to Bethany's comment, and thank you for the question, I think we are you know, and as many of our panelists have alluded to, we're only as strong as our partners out there, and that's dairy producers, that's other people working in the industry. And I think we have some tremendous networks. And I think the challenge for us as we move to this next phase and really become a formal research center is to really capitalize on all of those relationships and build strength that not only will support the things that we do from a research perspective, but really allow us to get our you know, the good word out there, all the results, and to also create I think a culture where we really welcome that input, those suggestions from industry partners, from producers about that will help us guide the research that we continue to do here as at the University of Guelph. So with that, I think we will wrap up today's session. A number of thank yous before we wrap up. Again, Dan Lacroix is our technical guru behind the scenes, and thank you, Dan, for all that you do for Dairy at Guelph. Thank you to our panelists this morning. I really appreciate the time that you put into this, both in preparing your presentations ahead of time and in the great discussion we've had here this morning. I think the hour and a half has flown by really quickly, and that's been because of the engagement. And finally, and probably most importantly, I want to thank our participants and our audience, again, for attending, for listening, for posting some great questions. And again, I think the gold star this morning goes to Harm Kelly, whose question I think evoked the biggest emotional response, I think, from most of our panelists. So again, thank you very much. Enjoy the rest of your day. And we hope to continue to connect with you on a regular basis. Thank you.